Well, hi everybody. This is lecture 7, I think. First part of chapter 13, pages 443 to 445 to 465-ish. We'll see how far we get in the textbook. And a word of warning, this is a long one, so bear with me. Probably going to go significantly over an hour, hopefully not over an hour and 15 minutes. But next lecture, the one you'll be watching for Wednesday, is significantly shorter, like really, really short. So I believe we're going to come in under 2 hours for the two lectures combined this week. So bear with a longer one, take the breaks you need to, pause it and come back at another time if that's necessary, and just know that you got a much shorter lecture coming on Wednesday, trying to keep things fair. So we're going to talk about today is starting off with the breaking down of sugars and fats, mostly sugars, and that's the catabolic side of metabolism, the breaking side of metabolism. That'll involve us just talking briefly about digestion and then going straight into glycolysis. Glycolysis literally means sugar splitting, glycolysis. So we're just going to split a sugar. As we split a sugar, we're going to break a whole lot of bonds and steal some electrons, and we're going to use some extra energy to make ATP as well. We'll take a side detour and talk a little bit about fermentation and why fermentation is done, and then we'll move on to the citric acid cycle, which is an electron stealing cycle. The whole purpose of the citric acid cycle is to steal electrons. And we'll close out by talking about how the citric acid cycle is the actual hub of all of metabolism and what we really mean by that. So, how do cells obtain energy from food? Well, the reason why we need energy from food is to bribe the universe. We've made this a central theme of this course. I'll continue to harp on this theme. It's a central theme of thermodynamics and biology in general. Life will not allow energy-consuming reactions to occur. If you're non-spontaneous and you have the universe is not going to let that reaction happen. And so the only way a highly ordered, low entropy state such as life can exist is if we are bribing the universe with released energy, exergonic reactions, and keeping the overall delta G's negative. We've talked about that a lot. Reactions must be spontaneous and free energy must be released if the low entropy state of life is going to be possible. And this energy that we're constantly bribing the universe with comes from food. It comes from the potential energy of the stretched out rubber band bonds, covalent bonds, of the foods we eat. The macromolecules in the foods we eat have high energy bonds with a great deal of energy of want in them. Those bonds are made of electrons, and when we break those bonds and steal those electrons, we're stealing that free energy, that potential energy of want, and then we can use some of that energy to bribe the universe. So it all comes down to these electrons. The electrons that make those bonds is where the energy lies. And if we want that energy, we got to break those bonds and steal those electrons. Well, sugars are the most common and readily available source of this energy. It's important to note that fats and proteins can be used in this way as well. Fats, in fact, have more bang for their buck. We talked about that in class. There's more energy per unit mass in fats than in sugars. They're just harder to metabolize. Proteins is kind of a macromolecule of last resort when it comes to energy production, but we can metabolize proteins and we can get energy or ATP out of them. It's worth noting that nucleic acids are a trivial source of energy. We do consume nucleic acids, RNA and DNA. They're in the foods we eat. Of course, we're eating things that were living. And so those living things had their own genome and had their own RNA. And we do break those nucleic acids down into their individual nucleotide building blocks. But we do that for anabolism. We take the nucleotides from the foods we ate and we use them to make our own new DNA and RNA. When we break those bonds, the rubber band covalent bonds of nucleic acids aren't very stretched out. So there's not a lot of potential energy of want there. There's not a lot of energy in those bonds, and so we really can't capture high energy electrons from them. Cellular respiration is the process by which electrons are harvested from these ingested macromolecules that we eat. Potential energy is released when we break those bonds by stealing those electrons, and then we take the energy from those electrons and use it to make ATP. If we think about glucose specifically, a 6-carbon sugar, the waste products of cellular respiration will be the carbons from glucose that we no longer need. We'll release them as fully oxidized carbon dioxide in our exhalate. The spent electrons, once we harvest those electrons and we milk every last drop of energy out of them to make ATP, the electrons still exist. They have just virtually no potential energy left in them, so they're garbage too. We got to get rid of them. We take those electrons and throw them onto oxygen. Reduced oxygen is water, and so we exhale that as well. And as we go through this process, in this video and much of next week, we're going to encounter what should be now an old friend quite a bit. NADH, our activated carrier for electrons, will play very big roles in these processes. You might remember from the last video that NADH is involved in harvesting electrons through catabolism. NADPH is our electron carrier for anabolism, but since we're going to be talking about catabolic processes, we're going to be talking about NADH a lot, our activated carrier of high energy electrons. So, let's begin. There's a lot of total free energy in the bonds of sugars and fats. Again, this is energy of want. It's potential energy. The covalent bonds in sugars and fats are fairly long. They're stretched. They want to snap like a rubber band, and that is potential energy. And when we break those bonds, just like breaking that rubber band, we get kinetic energy, snapping energy. By stealing the electrons of those bonds, we are stealing that potential energy, and then we can transfer it around. If all of the energy of these bonds was released at once, if we could somehow fully and completely metabolize a sugar or a fat all at once, the amount of energy released would literally be explosive. There's no way, there's no enzymatic process, there's no anabolic enzyme that has the ability to capture all of that energy in one shot. And so whatever would be capturable would be captured, and the rest of the energy would be released as enthalpy, as free energy, as heat. It would quite literally be explosive, and the cell would cook, would be killed by the heat released from this reaction. Not to mention, such an exergonic reaction would have a relatively high energy of activation, so high that it's unlikely that any enzyme would be efficient enough to lower that energy of activation to any feasible amount. So all of this, not good, not the way to do it. Instead, what cells do is they use a sequence of reactions. We're going to talk about a total of 18 reactions today, and each of these reactions plays a minor role, a sequential role. It's one step in the process of releasing this energy. Each of those reactions has a relatively low energy of activation that an enzyme can overcome, and each of these reactions releases a small amount of energy that can be efficiently captured and doesn't cook the cell. So this is a much more efficient way of releasing the energy from glucose, from any macromolecule. Most of the energy that is released by each of these sequential steps is captured either as electrons and put onto NADH, or captured as energized phosphate, and our activated carrier for phosphates is ATP. We know that from the last video. And when we do make ATP in this way, we're doing a process called substrate level phosphorylation. Substrate level phosphorylation occurs when a single reaction releases a good amount of free energy. It's got a pretty high negative delta G, and that free energy is used to phosphorylate adenosine diphosphate, ADP, to make it adenosine triphosphate. So in substrate level phosphorylation, we're talking about single exergonic reactions that are releasing enough energy to phosphorylate a single ATP. We're basically making ATP one at a time. That's to be contrasted with oxidative phosphorylation. Oxidative phosphorylation occurs in the mitochondria, and this is where we use the energy of oxidation, the energy of electrons, right? That's what an oxidation reaction is. It's the theft of electrons. We use the energy of electrons to make lots and lots of ATP all at once. We're going to talk all about oxidative phosphorylation next week, but I just want to introduce substrate level phosphorylation, making one or two ATPs, one or two at a time with a single reaction, as opposed to oxidative phosphorylation, basically using all of the captured energy from electrons to make a whole lot of ATP at once. All right, so let's eat something, shall we? Let's start with digestion. Ultimately, sugars, fats, proteins, they all are going to have their electrons stolen in the same way so that their free energy can be stolen and ultimately be used to make ATP. But since these macromolecules are so different, they have to be prepared for electron harvesting in different ways. And that, of course, is catabolic. It's the breaking down of large polymers into their individual monomers. Proteins need to have their peptide bonds broken and converted into individual monomers of amino acids. There are peptidases and other enzymes that break peptide bonds. Polysaccharides need to have their glycosidic bonds broken, and then individual monomers of monosaccharides are released. And fats have to have their covalent bonds broken so that fatty acids and glycerol can be released for metabolism. Again, we are breaking the phosphodiester bonds of DNA and RNA as well. We're releasing monomers from those so that we can reuse and recycle those monomers, but very little energy in those bonds. So this isn't really a nutritional source of energy. This is considered stage one of catabolism, the breaking down of large, complicated macromolecule polymers into their individual monomers. And this is really what we mean by digestion. This is what occurs in the small intestine. At the cellular level, this is what occurs in the lysosome. It's digestion, breaking big molecules down into small molecules. Stage two of catabolism is glycolysis. Again, literally translating to sugar splitting. Glycolysis occurs in the cytoplasm, and it's a chain of reactions, 10 reactions in fact, that split 6-carbon glucose into 2 3-carbon pyruvates. So we're not going to lose any carbon here. We're just taking the 6 carbons of glucose and basically splitting it into 2 3-carbon pyruvates. All the other macromolecules that we release from amino acids and from fats can also be catabolized by glycolysis or processes that come after. But in order to do that, they would have to be first converted into glucose or one of the other intermediates of glycolysis. We're going to see in a little bit fructose 6-phosphate's an intermediate of glycolysis. So if you're a macromolecule and you want to go through glycolysis, you're going to have to become glucose, glucose 1-phosphate, glucose 6-phosphate, fructose 6-phosphate, something like that, something that can enter one of the reactions of glycolysis. Again, glycolysis is cytosolic, it's occurring in the cytoplasm, and we're going to see as we go through this process, the outcome of glycolysis is not only 2 pyruvates, but also a profit of 2 molecules of ATP and a profit of 2 molecules of NADH as well. We steal energy in glycolysis, and we use some of that energy to phosphorylate ATP and some of that energy to activate NADH. Once pyruvate is made, it's transported right into the mitochondria through specific transport proteins, and the rest of cellular respiration is mitochondrial. In the mitochondria, pyruvate will be converted into acetyl-CoA. That acetyl-CoA will then enter the citric acid cycle, and we'll take it from there. Got rid of the nitrogen group, right? Urine, essentially, is nitrogen waste. So we get rid of that right away. You can't use ammonia. We don't want ammonia. Ammonia is not good for us. So we strip the amino groups off of the amino acids, and we take that carbon skeleton that's left over, and we can feed it into this process as well. So fats and the deaminated carbon skeletons of amino acids are also converted into acetyl, and they can be linked to CoA to make acetyl-CoA, and they can also be fed into the citric acid cycle. Acetyl-CoA is made in the hollow area called the matrix of the mitochondria. Once we make acetyl-CoA, again, we're ready for stage 3. Stage 3 is the citric acid cycle. The citric acid cycle is entirely mitochondrial, and it is nothing more than an electron-stealing apparatus. It's 8 reactions long. It is a cycle, so we go back to the beginning, and the whole purpose of the citric acid cycle is to collect redox reactions. Break bonds, steal electrons, because that's where the energy is. By the end of the citric acid cycle, all of the carbons of glucose will already be released as carbon dioxide, and all of the potential energy from the bonds of glucose will have been stolen or released and loaded onto NADH, our catabolic electron carrier. Yes, we'll make some ATP, not much, but we'll make some. Yes, we'll reduce one FAD to FADH2, but the vast majority of the energy that glucose has to offer will be loaded onto NADH by the end of the citric acid cycle. Now, getting ahead of ourselves just a little bit, but telling the story that's going to continue next week, once those electrons are harvested, mostly on NADH, they're going to be shuttled over to the electron transport chain. Little by little, passage by passage, redox reaction by redox reaction, that potential energy will be released, and it's going to be used for active transport. That energy from those electrons is going to be used to pump something against their concentration gradient. What's going to be pumped? Protons, hydrogen ions. Protons are going to be pumped against their concentration gradient using the energy from the electrons from glucose. And then those protons, with that massive concentration gradient, that ton of potential energy, those protons want to flow so badly, so there's so much potential energy there, those protons will be allowed to flow, and that kinetic energy of proton flowing is the energy that will be directly used to make ATP. That is the oxidative phosphorylation step. And again, I'm getting ahead of myself. So the electron transport chain and oxphos and ATP generation is all going to be what we talk about next week. We're going to focus today on the citric acid cycle and glycolysis. Well, the other way around, on the glycolysis and then the citric acid cycle. So at the end of this story of cellular respiration, the potential energy from the bonds of ingested macromolecules, specifically glucose, that the bonds are broken, the electrons are stolen, and that potential energy is transferred directly into the potential energy of three phosphates being stuck very close together and desperately wanting to get away from each other. That's ATP. And ATP is the universal energy currency molecule of the cell. No other molecule is so widely used by so many different reactions, different enzymes, in order to drive the reactions of life forward. When we make ATP, we're making on-demand usable energy that can be widely dispersed and keep life going. Actually, we're making a molecule that can be used to bribe the universe and keep reactions at a negative delta G, just in the way that we talked about in the last video, through those coupled reactions, those two-phase reactions where phosphates are being transferred. This entire process of cellular respiration extracts and captures and converts 50% of the available potential energy that glucose had to offer. And although that sounds pretty poor, keep in mind that the most current, modern, and economical car engines extract about 20% of the available potential energy. They make it. Right? It looks so innocuous. This is cellular respiration, as simply and as distilled as you can view it. Food and oxygen become ATP, reduced NADH, carbon dioxide is waste, and water is waste. That's cellular respiration. But how we get there requires 18 independent biochemical reactions plus the ETC and plus oxidative phosphorylation. It's quite the story. So let's tell it. Glycolysis, getting energy and making ATP from glucose, getting energy and making pyruvate from glucose. Again, glycolysis is completely cytosolic. All of these reactions will occur in the cytoplasm, and it's anaerobic. It doesn't need oxygen, so we can do glycolysis even in an oxygen-poor environment. Many biochemists and evolutionary biologists believe that for these two reasons, because it doesn't require an organelle and it doesn't require oxygen, glycolysis was probably the first catabolic pathway to evolve that still persists now in living cells. And again, one molecule of glucose containing 6 carbons is going to be split into 2 molecules of pyruvate containing 3 carbons each. We won't lose any carbons in pyruvate directly. But we're breaking bonds, so we'll steal some electrons, we'll release some energy, we'll use some of that energy to make ATP, and we'll make the most of it we can. In glycolysis, 2 ATPs are actually used up in the first 3 reactions, in reaction 1 and reaction 3, but we make 4 ATP molecules later on down the line. And so glycolysis yields a net profit of 2 ATPs, because we spend 2 early and then we make 4 later on. So we're walking away with a profit of 2 ATPs. And we're going to go into some detail here for glycolysis. For those of you with amazing retention that remember glycolysis from Bio 107, we're going to see it in much, much more detail here. So I want to be very clear for you what I'm going to expect you to know as we go into mastery checks and e-reports not too long from now. I do not require you to memorize the names of the enzymes. If you take biochemistry, that very well might be required. If you go to grad school, definitely you'll have to know all of these enzymes and more. But what I insist that you know are the types of reactions that are occurring in each step. And those two go hand in hand. You'll see what I mean in just a second. I also want you to know which steps use ATP and why ATP is being used, which steps make ATP and how ATP was made, and then where electrons are harvested. So that's what I want you to know from both the citric acid cycle as well as glycolysis. The type of enzyme and the type of reaction, and again, the two are largely the same, And is ATP being used and why? Is ATP being made and how? And what's going on with the electrons? You do not need to know the individual enzyme names, but if you do, you'll be setting yourself up for that much more success because the enzyme names are very descriptive, completely descriptive, and it'll help you remember the reaction types if you remember the names. But again, it's up to you. Glycolysis is not a cycle, it's a linear pathway, does not regenerate, and it's made up of 10 steps, all catalyzed by enzymes, of course. And panel 13.1 in the textbook, if you're working with the 6th edition, that's going to be on page 456 to 457, shows these reactions in wonderful detail. Textbooks does such a good job with aerobic respiration, and those are the images I'm going to be using on the next few slides. As a quick outline, the first 3 reactions of glycolysis are considered the energy investment phase. That's where we're going to use those 2 ATPs to energize and activate glucose. Then we're going to move on to the cleavage phase. This is where what was once glucose is going to be cut, and we're going to end up with 2 3-carbon molecules. And then the last portion of glycolysis is going to be the energy recoup phase or energy generation phase, where we're going to make those 4 ATPs and ultimately create pyruvate along the way. And again, if we burn 2 ATPs and we make 4, there's our profit. Also some useful resources. Table 13.1 in your textbook, regardless of which edition you're using, it'll be table 13.1, goes through the reaction types so that you're familiar with them. We're going to see kinase reactions. As a reminder, kinase reactions is when we add a phosphate group to a target molecule. We're going to see isomerization reactions, which is when a molecule is rearranged. We're going to see a mutase, I shouldn't say plural, we're going to see one mutase reaction, which is when a functional group is rearranged, not the whole molecule, but a functional group is moved, and the mutase reaction we'll see will be moving a phosphate group. And then we have dehydrogenase reactions. Now think about that for a second. Dehydrogenase. So this sounds like a reaction that's losing hydrogen. But if you're losing hydrogen, what else are you losing? Remember, follow the hydrogens. Any molecule that is losing hydrogens lost those hydrogens because it lost electrons. So these dehydrogenase reactions, they're actually oxidation reactions. These are where the electrons will be stolen. So we'll keep that in mind. Textbook lays it out so that the enzyme names are in yellow and the whatever is being changed in the substrate molecule to product is highlighted in blue. And they're going to use that symbology or that format not only for glycolysis but also for the citric acid cycle, so it makes it really convenient. So step 1, the enzyme's name is hexokinase. So it's all in the name. Hex is the prefix for 6. So a hexose is a 6-carbon sugar, like glucose. And we know what a kinase is. We certainly should know what a kinase is by now. It's an enzyme that has a phosphate group. So this is a phosphorylation reaction. Hexokinase is going to phosphorylate glucose and create a product called glucose 6-phosphate, just meaning that the phosphate was added to the 6th carbon. So first step, activation step. We're adding energy to glucose. We're energizing glucose by adding a phosphate to it. Phosphorylation reaction, catalyzed by a kinase. Step 2, the enzyme's name is phosphoglucose. Well, that doesn't surprise me. Phosphoglucose, phosphorylated glucose. That's what we just made, glucose 6-phosphate. Phosphoglucose isomerase. So this is going to be a rearrangement reaction. This is going to be an isomerization reaction. Glucose 6-phosphate is isomerized into fructose 6-phosphate. Bonds are broken and bonds are made, but no real energy release here, which is why we see the double arrow in the reaction. This reaction could be at equilibrium. There's no real energy change. So isomerization reaction, rearrangement. Glucose 6-phosphate becomes fructose 6-phosphate. And then step 3, another energy investment. We're going to energize this molecule even more with another kinase reaction, phosphofructokinase, PFK. Love that name, phosphofructo. So this is a kinase whose substrate is phosphorylated fructose. And what is phosphofructokinase doing? Adding another phosphate. It's a kinase. So this gives us fructose 1,6-bisphosphate. This means it's a fructose that has 2 phosphates on it, but the bis means that the 2 phosphates are in different places, and the 1,6, hopefully by now, is self-explanatory. Fructose 1,6-bisphosphate, fructose with a phosphate on carbon number 1, and a phosphate on carbon number 2. It's not a diphosphate. Phosphates are on different functional groups. So fructose, phosphofructokinase adds a second phospho group. Now, what I want us to appreciate here for just a second is look at this product molecule. We've got a big, highly negative phosphate here on one end of fructose, and a big, highly negative phosphate here on the other end of fructose. These two phosphates are both negatively charged. They want to get as far away from each other as possible, and they're stuck on either ends of fructose. This is creating an enormous strain on the fructose sugar ring. It's being pulled in two opposite directions at the same time. Well, what often happens to fragile things that are pulled in opposite directions at the same time? They split. So the next reaction, reaction number 4, is the split reaction. It's a cleavage reaction, and fructose 1,6-bisphosphate is cut. It's cleaved. Aldolase is the name of this enzyme. I won't go into why it's called that. It's not going to be very helpful. And again, you don't need to know the enzyme names, but certainly know that step 4 is the cleavage reaction. And this gives rise to two different molecules. One is referred to affectionately as DHAP, dihydroxyacetone phosphate, and the other we call GAP, glyceraldehyde 3-phosphate. Some people call that G3P. You may see that in other courses, but I've learned it as GAP and it rolls off the tongue better. So glyceraldehyde phosphate. So to summarize, fructose 1,6-bisphosphate with all that strain being pulled in opposite directions splits, it breaks. Aldolase cleaves it into DHAP and GAP. Step 5. Well, it would be pretty inefficient to have to treat DHAP and GAP separately. They're separate molecules, so they would need separate enzymes to continue on and become pyruvate independently. So here in step 5, the cell does something really clever. It does another isomerization reaction, and it isomerizes, or it rearranges, DHAP into a second GAP molecule. And this allows the cell to do everything just once, right? Efficiency. If it's really good at turning GAP into pyruvate, then let's just isomerize DHAP into GAP and keep that efficiency, rather than having to evolve a completely different biochemical linear pathway to turn DHAP into pyruvate. So step 5 is the isomerization of DHAP into GAP. And I just want to remind you that in step 4, when we split fructose 1,6-bisphosphate into one GAP molecule and one DHAP molecule, and now we've isomerized that DHAP into a second GAP, that means everything we talk about from this point on is going to be done in parallel. It's going to be done 2 times over for every molecule of glucose. So step 6, our first dehydrogenase. We're going to steal hydrogens, which means we are also stealing electrons. There's NADH being reduced. So GAP, and remember there's 2 of them now, so this reaction is happening twice over in parallel. GAP plus oxidized NADH and a phosphate is going to be dehydrogenated into 1,3-bisphosphoglycerate, 1,3-BPG. We're going to reduce that NADH, and there's so much energy in GAP that when we break that bond and steal that energy, we have enough energy to activate NADH through reduction, and still enough energy to left over to take a phosphate, a free phosphate, and add it to the oxygen that you see there in blue. And still enough energy left over that we have a negative delta G. So this is a very energetic reaction. We break this bond right here. Well, the glyceraldehyde 3-phosphate dehydrogenase breaks it, but we break this bond right here. And there's so much energy in these electrons that we can take some of that energy to put the electrons on NAD+, giving us NADH, and we can take some of that leftover energy and take this free phosphate and pop it onto that carbon, even though that requires energy, and still this reaction has a negative delta G. It's amazing. You can see 1,3-bisphosphoglycerate is a double phosphorylated molecule, just like the bisphosphate we saw in fructose. These 2 phosphates want to get very, very far away from each other. They're both negative, but unfortunately, phosphoglycerate is not a fragile molecule, so this molecule doesn't split. Instead, we are going to let one of those phosphates go. And just a reminder, all of this is happening twice over. So when we let that phosphate go, we're going to take that released phosphate and all the energy that comes with it and use it for substrate level phosphorylation. We're going to take ADP and phosphorylate it into ATP. Again, these 2 phosphates were under an enormous amount of strain. They wanted to get away from each other. There's a lot of potential energy of want, both in this bond and in this bond, but also in the repulsion of these 2 phosphates. A lot of potential energy. And that potential energy is sufficient enough that when we release this phosphate, we can take that released energy and use it to phosphorylate ATP. So 1,3-bisphosphoglycerate becomes 3-phosphoglycerate. And here we have a kinase because what's being phosphorylated is ATP, step 7. ATP is being phosphorylated, so phosphoglycerate kinase. And again, this is an example of substrate level phosphorylation. This one-off, a high energy reaction that allows us to make 1 ATP. Step 8, here's our mutase reaction. I told you there was going to be one of them, and I even gave you a heads up. We're just going to be moving a phosphate. Can't really call this an isomerization because the whole molecule isn't being rearranged. Glycerate is staying glycerate. So this is a mutase reaction. A functional group is being moved, and the functional group being moved is the phosphate. 3-phosphoglycerate, so glycerate with a phosphate on its third carbon, becomes 2-phosphoglycerate, or glycerate with the phosphate on its second carbon. And that's step 8. Step 9 is catalyzed by enolase. Again, more of these historical names. I won't go into it here, but if you'd like to know why it's named enolase, let me know. We can talk about it. This is going to be a dehydration reaction. We're going to pull a water off of 2-phosphoglycerate, giving rise to phosphoenol pyruvate. And for the first time, we see what we're after, PEP. By removing 2 hydrogens and an oxygen from 2PG, 2-phosphoglycerate, we make phosphoenol pyruvate. And then step 10 is the last step. We just pop that phosphate off, and we use that phosphate to do another round of substrate level phosphorylation. We make ATP, and we're left with pyruvate at the end of step 10. The enzyme that catalyzes this is called pyruvate kinase, and that's actually the name for the opposite reaction, right? If I read that pyruvate kinase, I'm an enzyme that catalyzes phosphorylates pyruvate. Well, that actually describes the reverse reaction. We're dephosphorylating pyruvate, but still gives you a hint for what it does. And it's the only enzyme name with pyruvate in it, and pyruvate's being made by step 10. So yeah, another substrate level phosphorylation example here as well. And we've made pyruvate. We've accomplished the goal of glycolysis. So again, in a nutshell, the 10 cycles, or 10 steps, of pyruvate allow us to make 2 ATPs, these 2 ATPs. We burn 2 ATPs, one in step 1 and one in step 3, but we make 4 ATPs because as soon as we split fructose 1,6-bisphosphate into 2 molecules of GAP, as soon as we do that split, we're processing those 2 molecules of GAP in parallel, and we make 2 molecules of ATP from each of them. So these 2 ATP molecules, they basically cancel out the investment that we made in step 1 and step 3, and then the other 2 ATPs are profit. And they were all these ATPs were made by substrate level phosphorylation, the transfer of a phosphate from a donor molecule that had a high energy bond to a recipient, which in this case was always adenosine diphosphate. And let's not forget, we've also reduced 2 NADHs, so we stole some electrons. And those electrons will go off to the electron transport chain, which is in the mitochondria, which we'll talk about next week. So before we move on to the citric acid cycle and talk about what happens to that pyruvate, let's talk just a little bit about fermentation, because fermentation is how cells produce ATP anaerobically, and it relies on glycolysis. For those of you that had me for Bio 117 for the General Biology 1 lab, hopefully some of this rings a bell because I tried to hit fermentation pretty hard with you all, and the lab report that you wrote in that semester was a fermentation lab report. So hopefully some bells are rung deep in the subconscious memory there from first semester. Glycolysis is typically followed by the citric acid cycle, the electron transport chain, and oxidative phosphorylation. That is by far, hands down, the most efficient way to extract the energy from these electrons, but it is an aerobic process. It requires oxygen. If we don't have oxygen, we don't have a place to put the electrons when we're done with them, and then the whole system backs up and becomes undoable. In the absence of oxygen, cells would die unless they can keep glycolysis going. And lots of cells can do this. There are fermentive bacterial cells, there's fermentive yeast, and even our own muscle cells can ferment for quite some time when they're deprived of oxygen before they begin to die. And so these cells are surviving on that measly little 2 ATP profit that comes out of glycolysis. But there's a big but here. The big but here is that glycolysis alone reduces NAD plus to NADH. And if glycolysis were to continue and continue and continue anaerobically, we would keep making NADH, but without oxygen around, we have no place to put those electrons. And so you can envision that after some time, all of the available oxidized NAD plus would be reduced to NADH by glycolysis running and running and running with no oxygen. All of that NADH would stay as NADH because the electron transport chain isn't accepting new electrons, there's no oxygen around. And once that last NAD plus molecule is activated and reduced to NADH, glycolysis would shut down. The reactions of glycolysis require dehydrogenation, and without NADH to accept those electrons and that hydrogen, glycolysis would shut down. So the 2 ATP profit of glycolysis could support life, but for that to be life supportive, glycolysis must keep running, and for glycolysis to keep running, we must regenerate NAD plus. We have to oxidize NADH back to NAD plus so that glycolysis can continue to run. Again, otherwise, over time, all of the oxidized NAD plus would become NADH, and glycolysis would shut down. It would have no place to put those electrons, and cells would die. Now, there's no source of ATP, and cells would die. So fermentation isn't energy producing itself. Fermentation is, regardless of the cell type, a series of side reactions that just gives the electrons on NADH a place to go. Those electrons are high energy, they're valuable, but without the ETC, we can't get that energy out. So fermentation just offloads and throws those electrons away as trash so that glycolysis can keep running and cells can survive on that 2 ATP profit. Now, if we're ethanol fermentation, alcoholic fermentation, like Saccharomyces cerevisiae or like some bacteria, what we're doing in the fermentation cycle is we've made pyruvate, we take the electrons from NADH and we use them to reduce pyruvate. That oxidizes NADH back to NAD+. There's the regeneration. Now glycolysis can continue. And a few reactions overlooked here, reduced pyruvate becomes ethanol. And that's the ethanol that's produced in fermented spirits, like wine and beer, and even distilled spirits are first fermented. So that's the ethanol that we drink as alcohol. But the purpose of that was not to make ethanol. The purpose of that, from the cell's perspective, was to just dump the electrons on NADH somewhere so that NAD plus could be regenerated and glycolysis can continue, because it's glycolysis that's keeping the cells alive. In lactic acid fermentation, it's a different set of side reactions that convert pyruvate into lactic acid, and all the other fermentation pathways is basically the same idea. Use pyruvate as an electron receptacle, turn it into something that's trash and will release as garbage, and get NAD plus regenerated so that glycolysis can continue. I also, before we go into the citric acid cycle, want to talk about things other than glucose and what they can become. Once pyruvate is produced in glycolysis, it's directly transported into the mitochondria, and in the mitochondria it's converted into acetyl, and that acetyl is linked to CoA. That 3-carbon pyruvate, to make that possible, is decarboxylated, and there's a big word, decarboxylated, to make 2-carbon acetyl. So acetyl is a 2-carbon molecule, pyruvate of course has 3 molecules. If you want to go from 3 carbons to 2 carbons, you got to decarboxylate, you got to lose a carbon. And when you lose a carbon here, you're actually losing it as carbon dioxide. So this is one of the carbons from glucose that's being released as carbon dioxide. And I want to again emphasize that 2 carbon dioxides are being released here per glucose. Because if we've metabolized glucose, we've made 2 pyruvates, we've used those 2 pyruvates to make 2 acetyls, which means we did 2 decarboxylations. So 2 of glucose's carbons are lost here as we do these reactions in parallel. And to release that carbon dioxide, we broke some bonds. That means we had the opportunity to steal some electrons, and steal electrons we did. We made NADH along the way as well. This conversion of pyruvate into acetyl through decarboxylation and oxidation is catalyzed by the pyruvate dehydrogenase complex. There it is again, dehydrogenase, stealing protons, but really so that we can steal electrons. We refer to this enzyme as the PDC, and it is a doozy. It's made up of 3 separate complexes that are stuck together. It's even structurally beautiful. It looks like a giant cube. We won't go into any detail on this, unfortunately. It's just beyond the scope of this class. But depending on how biochemistry is taught, they might go into some detail on the PDC. And if you ever go to grad school, like well, I shouldn't say true grad school, because medical school is true grad school, but like a doctorate or a master's degree, you take heavy duty graduate level biochemistry, you'll learn all about the PDC there as well. For our purposes, pyruvate goes into the PDC. It's got 3 carbons. It's decarboxylated, giving us 2 carbons, and then those 2 carbon intermediates are ultimately converted into acetyl. This is acetyl right here, and then acetyl is linked to CoA, making acetyl-CoA. Then that acetyl-CoA is fed directly into the citric acid cycle. In the citric acid cycle, acetyl is joined to an intermediate called oxaloacetate. Oxaloacetate is a 4-carbon molecule. Acetyl has 2 carbons. And when acetyl joins onto oxaloacetate, you make 6-carbon citrate, which is where the citric acid cycle gets its name. But we're going to talk about that in just a little bit. As I said, I want to tell you about other things that become pyruvate or acetyl, and that includes fats and proteins and amino acids. Fats are purely hydrocarbons, right? They're so hydrophobic. That's why they're hydrophobic. And they can be directly catabolized or broken down into acetyl. And there it is. That is beta oxidation. I don't want you to know this. You don't have to know this. Let's just look at a few key things so you get just a whiff of it, a taste of it. Here's a triacylglyceride, right? This is fat. This is the fat that keeps an animal living through the winter. Long-term energy storage. Glycerol is the clothespin holding everything together, and we've got 3 acyl groups, 3 fatty acids. Where's my pointer? 3 fatty acids coming off of that glycerol. Triacylglyceride. We're going to first break the bonds that hold those acyl groups to the glycerol and release the fatty acids themselves. So here's the fatty acid being released. A lot of energy in that fatty acid. That energy has to be carried, and it's carried by CoA as an activated carrier. Then that fatty acid is going to be delivered by CoA into beta oxidation, a beta oxidation cycle. And this is what we're seeing here. This is beta oxidation. And just to point out, just to have a sense of it, in beta oxidation, electrons are being stolen onto FAD, and quickly they're transferred to NAD. Electrons are being stolen and put on NAD. And then ultimately, as we do that again and again and again, hydrocarbon by hydrocarbon by hydrocarbon, we make acetyl, and we link that acetyl to CoA. So even in beta oxidation, the breakdown of fats, we're breaking bonds, stealing electrons, and ultimately making acetyl. Same general premise as what we just saw for glucose. And again, many amino acids can also be broken down and turned into either pyruvate or acetyl-CoA. And so this makes the citric acid cycle the hub of breakdown, of catabolism. It's also the hub of anabolism, which we'll talk about in just a little while near the end of this lecture, and you'll go into an enormous amount of detail on anabolism if you take biochemistry. But for our purposes, we're going to talk about the citric acid cycle catabolically, meaning we're breaking bonds, stealing electrons, and reducing NADH. Again, the citric acid cycle starts with oxaloacetate. It's 8 reactions in length, and it ends with oxaloacetate. This is why it's a cycle. Glycolysis started with glucose and ended with pyruvate. It is linear. The citric acid cycle starts with oxaloacetate and ends with oxaloacetate, and making it a cycle. Again, 8 steps in this reaction, and we'll talk about these steps in detail, but you can see right away, we broke bonds and stole electrons, broke bonds and stole electrons, broke bonds and stole electrons, broke bonds and stole electrons. That's the purpose of this cycle. It's an electron stealing cycle. We make 1 triphosphate along the way just because we can, there's enough energy to, but the purpose of the citric acid cycle is to break bonds and steal electrons because that's where the energy is. Okay. Again, the citric acid cycle is mitochondrial. It's occurring in the mitochondrial matrix, and it will ultimately completely oxidize all of the carbons, all 6 carbons from glucose, stealing electrons from those carbons and then releasing the carbons as carbon dioxide. By the end of the citric acid cycle, there will be no more atomic energy. All of the energy will be on the electrons that are captured and about to go to the electron transport chain. By the end of the citric acid cycle, all of glucose will have been decarboxylated and released as carbon dioxide. So we can see that here too. Remember, we're doing 2 citric acid cycles for every glucose we consume because we made 2 acetyls from that glucose, because we made 2 pyruvates from that glucose. And we already lost 2 carbons when we converted pyruvate into acetyl. So here we see a carbon dioxide comes off here in step 3, and a second carbon dioxide comes out here in step 4. So 2 carbon dioxides coming off of this citric acid cycle, 2 more coming off of the other acetyl that we made from that glucose, so there's 4. And then we saw 2 carbons released as carbon dioxide when we converted pyruvate into acetyl. There's your 6 carbons, there's glucose. So by the end of the citric acid cycle, glucose is exhaled, and all of the energy glucose had to offer is stolen on the electron carriers that we make. Yep. And then those electrons are being carried by the activated carrier NADH, the exception, 1 pair of electrons carried by FADH2, and those will be shuttled over to the ETC and used for making ATP. So the citric acid cycle is presented in its own panel, pages 460 to 461 if you're working off the 6th edition. And again, same symbology. Enzyme name in yellow and what's changing, what's being moved in blue. So step 1, here's oxaloacetate. In comes acetyl being carried by CoA. CoA is cleaved away from the acetyl. That CoA will be released and it can go off and pick up another acetyl group from the PDC. And the acetyl group here is linked onto oxaloacetate in a 2-phase reaction, giving us citrate. And that gives the citric acid cycle its name. Citrate is a 6-carbon molecule. So acetyl is joined onto oxaloacetate and citrate is made. This is a ligation reaction. Step 2 is an isomerization reaction. Well, we know what that is now. Citrate is going to be rearranged and isomerized to make isocitrate. Isocitrate because it's an isomer of citrate. This is a really interesting reaction. You don't really have to know this, but in order to do that isomerization, the enzyme that catalyzes this, called aconitase, actually removes the water and then adds that water back, but adds that water back in a different place. And that's what allows the isomerization to occur. But yeah, we just switched the hydroxyl and the proton, and we made isocitrate. So it's an isomerization reaction. Step 3. Step 3 is our first oxidation reaction. Also a twofer. We're going to first break some bonds, steal some electrons, and reduce NADH. And then we're going to decarboxylate the intermediate, turning isocitrate into alpha-ketoglutarate, or alpha-KG. The enzyme responsible for this is isocitrate dehydrogenase. Talk about a perfect name, right? Isocitrate, what it's working on. Dehydrogenase, what it's doing. And by stealing protons, we're stealing electrons. Our first oxidation reaction here. Oxidation of the substrate, reduction of NADH. So it's a redox reaction. Electrons are being transferred, electrons are being stolen, and NADH is made. Step 4. Why stop a good thing when it's working? Do it again. Alpha-ketoglutarate dehydrogenase is going to do that. It's going to oxidize alpha-ketoglutarate, break some bonds, steal some protons, steal some electrons, and use those electrons to reduce NADH. And we broke bonds, so we're going to release that last carbon from glucose. Like boom, boom, right? Reduction of NADH. Oxidation of the substrate. And by the end of this step, step 4, we've completely decarboxylated glucose, and all of glucose is now released as carbon dioxide. Product of this reaction is succinyl-CoA. High energy molecule linked onto CoA. CoA is carrying succinyl. Another redox reaction, electron stolen, NADH is reduced. Step 5, succinyl-CoA synthetase. This is going to take the high energy bond between CoA and succinyl, break that bond, and use that bond for a phosphorylation reaction. We're going to phosphorylate GDP, guanosine diphosphate, into GTP. Most of the time, very quickly after GTP is made, the energy of that third phosphate is used to turn ADP into ATP. So in other words, GTP gives that phosphate to ATP right away, but a triphosphate is made. The only time we make a triphosphate in the citric acid cycle. And the end result of this is succinate. So again, the high energy bond that's linking CoA to succinyl is replaced by a phosphate briefly. Then that high energy bond is broken and used, the energy released is used to phosphorylate GDP into GTP, our first triphosphate, and succinate is made. Step 6. Succinate is going to be oxidized to make fumarate, and another great enzyme name, succinate dehydrogenase. This is going to take electrons and put it onto FADH2. And just put a pin in this, a paper clip in this, make a note in your notebook or whatever you're taking notes in. These electrons are going to stay on succinate dehydrogenase. They're not mobile. And succinate dehydrogenase is actually also called complex 2 of the electron transport chain. So it goes by 2 names in 2 different contexts. So just keep in mind that these electrons are stuck on FADH2. FADH2 is stuck on this enzyme, and this enzyme is going to be complex 2 in the ETC. But for now, broke some bonds, stole some protons, stole some electrons, did a dehydrogenase reaction. It's a redox reaction, of course, and the electrons were put onto FADH2. Fumarate is the product. Fumarate is going to be hydrated. A water is going to be added to fumarate to make malate. The enzyme is fumarase, so it's a hydration reaction. And then our very last reaction of the citric acid cycle, our last chance to steal electrons. Don't kid yourself. If we got one more chance to steal electrons, we're gonna. So this is going to be a redox reaction. We're going to break some bonds, steal some protons, steal some electrons, reduce NADH. This is malate dehydrogenase. Of course it is, because it's dehydrogenating malate, and that's going to regenerate oxaloacetate. Step 8 bringing us right back to step 1. Now this oxaloacetate can accept another acetyl if one is being delivered, and the process can repeat as a cycle. So this beautiful figure shows the citric acid cycle in all of its glory, as we just described it. And again, I just want you to know the type of reaction occurring in each step, what's going on with the energetics, what's going on with the electrons, what's going on with the triphosphate, that generation of GTP there in step 5. So to summarize all of this, per acetyl-CoA, so per one run of the citric acid cycle, 3 NADHs are reduced. That's a total of 6 electrons stolen. 1 FADH2 is reduced, so 2 more electrons there, and 1 triphosphate was made. And if we think of it in terms of glucose, because 2 citric acid cycles run for each glucose we metabolize, these numbers are doubled. So per glucose molecule, we're making 6 NADHs, 12 electrons there, 2 FADHs, 4 more electrons, and 2 triphosphates. So we got ourselves a lot of electrons. All of the energy that glucose had to offer is in those electrons, and all of those electrons will now go to the electron transport chain. Okay, so let's leave that there for now in terms of catabolism, but let's just take a few minutes to appreciate the role of the citric acid cycle in anabolism. We don't dive too deeply into anabolism here. Again, we'll leave that to the biochemists. But while we're talking about the citric acid cycle, it would actually be irresponsible to at least not mention it. Most anabolic pathways, most biochemical pathways that make the molecules we need, steal the precursors or building blocks of those biosynthetic pathways from either glycolysis or the citric acid cycle. So let's take a look here. So from glycolysis, glucose 6-phosphate, the very first thing we make in step 1 of glycolysis, well, that's a precursor for making nucleotides. Fructose 6-phosphate is used to make some amino acids, some glycolipids, some glycoproteins. DHAP, remember DHAP? We isomerize it to a second gap. Well, we don't have to do that. If we got plenty of ATP, plenty of pyruvate, we can take some of that DHAP and use it to make lipids. Phosphoenol pyruvate, the second to last product of glycolysis, is a precursor for making pyrimidines. Pyrimidines being cytosine and thymine, also some amino acids. We can make serine directly from 3-phosphoglycerate, alanine directly from pyruvate. Citrate can be used to make cholesterol and other fatty acids. Alpha-ketoglutarate from the citric acid cycle can be used to make glutamate and other amino acids, and the purines, adenine and guanine. We can make heme from succinyl-CoA, aspartate and other amino acids, purines and pyrimidines from oxaloacetate directly. It's incredible, right? Like, these are all of the things we need to survive, and they're being made directly from precursors that are stolen from this central spine of glucose metabolism. So it's for this reason that glycolysis and the citric acid cycle are considered to be the central hub of all eukaryotic metabolism. No matter what diagram you look at the total biochemistry that's occurring in a eukaryotic cell, this is the diagram from your textbook, a very, very stripped down impressionist view. Here's a slightly more accurate and more detailed diagram of eukaryotic metabolism, but right here down the center, glycolysis and the citric acid cycle. Right down the center, glycolysis and the citric acid cycle. And if you learn real metabolism, eukaryotic metabolism, you'll learn this table. If you do go on to grad school in the hard sciences, like for a master's or a PhD, you'll basically learn every pathway on this table. I think I knew, at some point, I think I knew nearly everything on this table. Certainly by the end of my 3rd year of my doctoral training, I had all this crap jammed in my head. I don't remember all of it anymore, unfortunately. But all the same, this is all of eukaryotic metabolism. If it's being broken down or being made in a eukaryotic cell, it's being represented on this table. And right at the center, everything either feeding into or coming out of the central spine. You can't see it, it's too small, but trust me, right here it says glucose. And glucose is metabolized into two pyruvates. Pyruvate right here by glycolysis. Pyruvate is converted into acetyl-CoA by the PDC, the pyruvate dehydrogenase complex. And this bad boy that you see right here, where'd my arrow go? This bad boy that you see right here is the citric acid cycle, right in the center of the diagram, because everything either comes out of these or feeds into these. The unifying, connecting, bridging hub of catabolism and anabolism is the citric acid cycle and glycolysis, the backbone of eukaryotic metabolism. So, let's set you up for next week. I know it's the Monday video. We're going to be going off in a slightly different direction for Wednesday. Again, a very short video. But let's set you up for what's coming up next week as we continue this story. Per molecule of glucose, we've converted one glucose into two pyruvates, converted those two pyruvates into two acetyls, and ultimately decarboxylated all of that and released it as carbon dioxide. Along the way, we made a total of 2 ATPs in glycolysis, 2 GTPs, because we ran the citric acid cycle twice. We can use those GTPs to make ATP if we need it. We've gotten 20 electrons on NADH, and we got 4 electrons on FADH2, right? So 10 reduced NADHs and 2 reduced FADH2s. And all of that was glycolysis in the cytoplasm and the citric acid cycle in the mitochondrial matrix. But we're not done. Next week, we're going to send all of those electrons on those electron carriers, those activated electron carriers, to the electron transport chain. They will drop their electrons off at the ETC, and there those electrons will be passed from carrier to carrier through a series of redox reactions, each of those redox reactions having a negative delta G, releasing a little bit of the potential energy from those electrons, and then that potential energy is going to be used to pump protons. All of this is happening along the inner membrane of the mitochondria, and it's across that membrane that the protons will be pumped. The pumping of these protons will create an enormous proton gradient with a huge amount of potential energy of want, and then those protons will be allowed to flow and make oxidative phosphorylation possible. OXPHOS is the production of lots and lots of ATP molecules through the kinetic energy of proton flow. So at the end of the ETC, when all of the potential energy has been released from those electrons through all of those redox reactions occurring in a chain, we're still going to have electrons. They're just going to be low energy. All the energy was already released. We're going to take those useless, empty, low energy electrons and use them to reduce oxygen. Remember, oxygen has a very high electronegativity. It loves electrons, so it'll even take low energy electrons. But wherever the electrons go, the protons follow. So if you're going to throw 2 electrons onto an oxygen, you're going to get 2 protons to go along with it. And so reduced oxygen becomes water. And that's the water that we exhale. That's metabolic water. So the reason why we exhale carbon dioxide and water is because the carbon dioxide in our exhalate is the spent carbons from glucose, and the water in our exhalate is the spent electrons from glucose. Amazing, right? So in the end, the oxygen we breathe in is nothing more than empty trash cans waiting to be filled with depleted, energy-empty electrons. And then when those trash cans are filled, they become water. But again, that's all for next week's videos. As we move into the second video for this week, again, it's going to be quite short, we'll discuss how glycolysis and energy storage are regulated and what cells do when they don't need glucose for this process because ATP levels are already high. So until then, to summarize what we did talk about here, cellular respiration is the process used by cells to extract energy from the macromolecules that we eat, converting the potential energy of the bonds of those macromolecules into the potential energy of activated ATP. Catabolism, or the breaking half of metabolism, involves 3 stages: digesting the polymer down into its monomers, and then feeding those monomers into either glycolysis or the citric acid cycle. If it's sugars, those sugars are going to be fed into glycolysis and then the citric acid cycle, and energy is going to be captured as triphosphates, reduced NADH, and reduced FADH2. Glycolysis splits glucose into two 3-carbon pyruvate molecules, produces a net profit of 2 ATPs as it does so, and 2 NADHs are reduced. All of that happening in the cytoplasm. Pyruvate's converted into acetyl-CoA in the mitochondria. That's done by the PDC, the pyruvate dehydrogenase complex. And acetyl has 2 carbons, pyruvate has 3, so that also is going to involve a decarboxylation event, releasing carbon dioxide. And we broke some bonds, so we steal some electrons. We'll reduce some more NADH. The citric acid cycle's job is to steal electrons, and that's what it does really, really well. The citric acid cycle will fully oxidize acetyl-CoA, releasing all of those empty carbons now as carbon dioxide, and make lots of NADH, make FADH2, electrons again are being harvested, and make a triphosphate in the form of guanosine triphosphate. And also it's the citric acid cycle, so we're going to regenerate our starting point oxaloacetate. The electron transport chain, or ETC, is going to use the electrons from NADH and FADH2 to pump protons. The potential energy from those electrons will be released. That energy will be used for the active transport of protons, and that's going to create a proton gradient. The potential energy of that gradient will be used for ATP synthesis. What we saw in this video was substrate level phosphorylation, where energy is being released by single reactions and energy is sufficient to add an additional third phosphate onto ADP, making ATP one at a time. We saw that in glycolysis and in the citric acid cycle. In the ETC, oxygen is going to act as the final electron acceptor, a trash bin for spent electrons, and then water will be the byproduct of reduced oxygen. And finally, kind of summing everything up, biosynthesis in general relies on the intermediates of these processes, making the citric acid cycle the hub of all of metabolism. Nearly everything the cell consumes is broken down in this way, in some way, reliant on the citric acid cycle, and nearly everything the cell makes steals precursors from these processes as well. So, I don't have my timer running. I'm actually recording this on my laptop, so I have no idea how long this video ended up being, but for however long it is, my apologies and my promise that the next video will be much shorter. As always, thanks so much for watching, and let me know whatever questions you have about this material. I'm guessing you're going to have some, and we'll talk about them in class.